not just the biodiversity, but almost all of the processes and biogeochemical cycles in the ocean. And they're largely, of course, impacted by humans. And you, in the past, couple past talks at the BioNoon, you have heard about the microplastics in the ocean, about the pollution and carbon cycling, for example. And so today I would like for us to focus on oxygen, one of the key molecules for the complex life on Earth. That's, you know, the life is largely dependent on oxygen to produce energy. And if we look at the last 5 decades, we will see that the levels of dissolved oxygen in the ocean have significantly dropped. And we are facing this expansion of these like anoxic areas or anoxic environments, and sometimes they are also called dead zones. But today I would want us to realize that these anoxic environments are in no means dead, and they're actually booming with life, or there is life thriving in these harsh conditions. And so apart from, you know, archaea and bacteria, which I'm absolutely certain all of you can distinguish from these two pictures, there is also complex life, like eukaryotes, including us, you know, like metazoans. So you see these, you see like the life in anoxia ranges from these single-celled organisms like different amoebae or flagellates that the lab that I'm coming from in Charles University in Prague is largely focused on, all the way to complex metazoans like Loricifera, or recently discussed cnidarians, actually here up in the right, Henneguia, that's actually parasitic, but I will talk about free-living organisms, and all of the others here in the pictures are free-living. So if you look at the eukaryotic tree of life, you will see that anaerobiosis has actually originated many times independently within eukaryotes. And I would like for us to realize that anoxic environments, or anoxia, is not only, you know, in deep ocean or in like ocean floor or sediments that are difficult to access, and we need all these like remote operated vehicles to sample these spots, but that actually anoxic environments are everywhere around us. Yeah, I actually live just by Point Judith, so there's amazing anoxic environment and salt marshes just behind the house here, and so we sample a lot of these, and we don't have to actually you know, use any robots or machines. We can just kind of dive in the mud, and sometimes it can also get dangerous, and you need someone to pull you out if you're getting stuck in the mud. But it's nothing like the deep sea robots. But so why we are sampling these sediments, or why I'm interested, are ciliates that are actually one of the most well-studied groups from protists, from single-celled eukaryotic organisms, and they're also masters in penetrating various even harsh or extreme environments, such as anoxia or oxygen-depleted environments. And ciliates are, you know, very important grazers and they play a huge key role in microbial food webs, and so do the anaerobic ones. And if we compare the known anaerobic diversity, because ciliates are very very diverse. They include currently about 14 classes of very morphologically, ecologically, and evolutionary diverse organisms. And so the anaerobic representatives are largely known, or one of the main groups are Metopida, and many other groups that I will today talk mostly about the Metopida that I have focused on during my PhD and now during my postdoc. And I want you to see that the diversity of anaerobes or anaerobic ciliates is by no means covered, and we keep discovering more and more new organisms, such as these deep sea Karyokotrichia. They were discovered in 2011, or pretty recently described class of odontostomata that were known from the past, but just 2 years ago, Fernandez finally published some molecular data, and that have led her to and her team to realize that it's a distinct class. And this year, finally, after 10 years, we have published organisms that we were studying for many years that are also 2 new distinct classes of ciliates. And contrary to deep sea Karyokotrichia, they are inhabiting shallow beach sediments or different brackish salt marshes like Point Judith. And so apart from the role of ciliates as predators or grazers, they're also very well known for being able to establish various relationships, and they can form symbiosis as symbionts themselves or as hosts. So obviously it's a very successful group, but if we think of why are they so successful, we can also think of it as why are they so friendly, because they really form symbiotic relationships, not just with eukaryotes or and bacteria, but also with archaea, and they are known to host as one of the few groups. They host intracellular archaea within their cells, which is actually the situation in anaerobic ciliates. And from early 90s or even before, we know that ciliates, and particularly metopids, can host methanogenic archaea within their cells, and they also host sulfate-reducing bacteria, mostly as ectosymbionts, such as the case in the Cariaco ciliates here up on the right. And so obviously symbioses are important to the ciliates and the prokaryotes themselves, but why are they important to us? And here I'm realizing I wanted to say, why are they important to us as like all humans, not just to the US, as it might seem here in the title. But anyway, so the methane or methanogenic archaea are producing methane. They're synthesizing methane thanks to the hydrogen that is being produced by the hydrogenosomes or mitochondria within the cells of its host, or of the ciliates. And obviously methane is one of the most potent greenhouse gases that plays a huge role in the climate change, and sulfate reducing bacteria are also utilizing the hydrogen to produce the toxic hydrogen sulfide. So you see that they're very important. They play important roles in major biogeochemical cycles. So I think we should keep learning more about them so we can understand these processes as well. And so if we go back to the processes that are happening, or I will get to the results, or sort of what I would like to cover from our studies today. And I have divided it into sort of 3 parts that are majorly overlapping, so it's not like 3 distinct parts. And one of them is the diversity of the free anaerobic ciliates. That was a big part of my PhD project, and it was kind of the basis from which we can build upon studying the evolution and the symbiosis in these anoxic environments. So to start with the first part, we have over the years we have mapped the diversity of symbionts, or of anaerobic ciliates worldwide. We have sampled freshwater, marine, and brackish habitats, and we have established the largest culture collection, which is interesting because they can also the ciliates are maintaining their symbionts there, which was quite difficult to establish in the past. And we have focused on the morphology and on the ultrastructure. You can see the dikinetids in transmission electron microscopy here, which are also important in assessing the ciliate taxonomy. And we have performed molecular characterizations, or we have analyzed molecular data that would tell us more about the phylogeny of these ciliates. And we have also created, in collaboration with this British initiative on the diversity of ciliates, this guide for ciliate taxonomy, because if anyone listening is involved in taxonomy of anything, you know that it's mostly huge chaos, and it's difficult to orient oneself. So a part of the study on the taxonomy or on the diversity are also is the formation of the cyst, because ciliates are using these in times where the conditions are even more harsh than just low oxygen. And we have seen them a lot in our cultures, and sometimes it's easy to get the ciliates and cyst, but it's difficult to get them to exist and kind of revive. But interestingly, we have found out that the symbionts, the methanogens that they are hosting, are actually also active during the cyst stage. You can see here is the autofluorescence that methanogens are typical for the blue signal and it's active within the cyst. So that is also telling us more about the mode of transmission, the vertical transmission of these symbionts. And I have been trying to see more about the structure of the cyst, because there is not many studies focusing on it, but I have found out this this like granular structure in the sort of like a plug that is enclosing these cysts, if you see it here. But I have no idea what could it be formed by. So if any one of you have any idea, please let me know. And so we have revised the taxonomy, and I will definitely not bore you with any, taxonomic results or phylogenetic trees, but I just wanted you to see how the tree looked like when we published our first paper, or when we started analyzing these. There were only several known species or several known molecularly characterized species, within Metaphyta, the group of my focus. And this is the latest current tree that we have been lucky to be able to expand largely, which again is making it easier for us to study the evolution and the anaerobiosis as a whole, and the nature of these symbioses. So if we look at another phylogenetic tree that's including all ciliates, you will see that the symbiosis has happened in basically, you will find some anaerobes in almost every major clade or lineage within ciliates. Some of them, these light blue, are lineages that are including either some discrete anaerobic species or microaerophilic species, or just microaerotolerant ciliates. And the dark blue are lineages that are actually only obligate anaerobes, and so all of the representatives are only anaerobic. And you see that's basically the whole diversity. And we have also discovered 2 new ciliate classes, which are here within the tree. You see, sorry, I have lost my mouse. You see here Parablepharisma and Muranotrichea. They are cosmopolite marine anaerobic protists or ciliates, and for anyone that's not studying ciliates, I'm sure they look exactly identical to you, but I wanted you to see that they're different in eyes of protistologists, or they have very many unique features. And here in the Muranotrichea, you can see this interesting file of membranes or of cilia that are covering their cytostome, their cell mouth, that they are using to filter bacteria. And these are the Parablepharisma. And if you look at the coat of the cell, or the surface of the cell here, either in the drawing or in the scanning electron microscopy, you can notice that they're covered by these rods. And if we look at more closely, you will see that there are 2 types of these rods. One of them is larger and thicker, and the other ones are smaller. Also, don't get confused by the cilia. All the long rods are the cilia. But that brings us to the second large part of my talk, which are the symbioses. And so obviously there are ciliates that are hosting multiple symbionts, and if we look at the overall diversity or the different groups, we will see that they host 2 major groups, the methanogens and sulfate reducing bacteria, but in different localizations, the symbioses are of different coevolutionary length, or I mean, they're some are more tied or specific, and some are more loose and recent. And we are trying to find out more about the coevolution and about the host specificity. And we were hoping it could tell us more about why are ciliates so keen to reside in anoxia, and could the symbionts play any role in this? So here you can see more about the symbionts of the Parablepharisma. When we first looked at the autofluorescence, we realized that these are actually ectosymbiotic metanogens, which has not been previously described in any free-living organisms. Their metanogens are always known as endosymbionts, because they're protected by the host cell from the outer environment, and also from predatory organisms or predators. So this is very unique case where the metanogens are on the outside of the cell, and they're accompanied by another type of symbiont, which we in fish found out or identified as delta proteobacteria that are sulfate reducing. So there is a lot of competition going on the surface of these ciliates. Here you can see that they're visible even in vivo cell. And if we look at the other group, the Symbionts of Muranotrichea are also of two types. We know that there are also these sulfate-reducing bacteria on the surface, although they look completely different. You see that they have this sort of specific arrangement in like following the kinetids, not forming the thick coat as in the Parablepharisma. And we also know that they host these endosymbionts inside, but we have tried to sequence them and apply different kinds of fish probes, and so far we just know that they're not metanogens and not sulfate reducing bacteria, but I would like to figure it out at some point. And if we look at the attachment of the symbiont cells, again, even in the Metopoda, you saw that in the unrelated distinct classes, they look differently, but even if we look inside of a single lineage, the Metopoda, you see that they are very diverse, and in terms of the morphology or attachment and localization on or within the cells, you see that they can be following the cell surface as these long rods, or they can form like a thick coat that's covering the entire cell. Like here you can see this protargol stained cell. And when it comes to the endosymbionts, we can find them frequently in the vicinity of the mitochondria, or as we call it in anaerobic organisms, the reduced mitochondria-related organelles. And it's because they're utilizing the hydrogen that is produced by these mitochondria to form the methane. And that brings me to kind of the first, when we wanted to know more about the nature of these symbioses, Roxanne has sequenced and analyzed the first genome of this endosymbiotic metanogen, which was shown to be Methanobacterium. And from looking at the genome, or generally at the symbiont, we found out that it's quite closely related to other free-living methanogens, but at the same time there are some changes that have happened during the evolution or the coevolution, although when compared to other typical endosymbionts in other protists or animals, it's different from the general pattern that we usually see, that the genome of the symbiont is usually significantly reduced, sometimes all the way to form an endosymbiotic organelle, such as the mitochondria, you know. So in the case of the ciliate, the methanobacterium genome is not that reduced as we could expect, which is something interesting to think about. Although we have found some genes that are likely involved in the symbiosis or in the transmission or transfer of proteins and other things like that. And I have mentioned that this symbiont of the freshwater metopid was Methanobacterium, but if you look at the 16S phylogenetic tree, you will see that the yellow ones are the symbionts or endosymbionts of anaerobic ciliates, and you see that they're scattered throughout the whole tree of Eukaryotes. So albeit there is not just, obviously there is not just one lineage of methanogens that would be able to form these symbioses. It's like a very common thing for them. And as we try to assess the host specificity, we same as the other authors in the 90s or later on have been trying to find out, are coming to these challenges that there is no clear pattern that would tell us more about the host specificity, because the closely related species are sometimes hosting unrelated methanogens or unrelated symbionts, but at the same time unrelated ciliate species, as you can see here. This KU10, that's Metopus S, unrelated to Rika or CSS, Metopus Heterometopus paliformis. It's still Metopus, but they're on different ends of the phylogenetic tree, and they're hosting very closely related symbiont, or in some cases they host the identical symbiont. But then again, to make it even more complicated, this does not apply always. Sometimes we also see a genus of the metopids that are all of the species within the genus are hosting the same methanogenic species, the symbiont. And then in other cases, it's not like that at all, and they host different species. So it's very complicated, and we're still trying to get a hold of it, and it's a major part of the plans for my postdoc here, and we are still collaborating on it with my supervisor with my past supervisor from my PhD, Ivan Čepička from the Charles University. And I would also like to find out more about the symbionts in the other anaerobic cilia groups, such as the newly discovered Muranotrichea and Parablepharisma, or even other unrelated lineages that we haven't gotten to even describe yet. So it's kind of leading us to realize that ciliates are actually great potential model group for studying these anaerobiosis or the origin of anaerobiosis and the life in anoxia in general, which we are also working on in cooperation with Ginny Edgecombe and Maria Pakiadaki and Fatma Goma from Woods Hole and Harvard. And that brings us to the third part of the talk, or the last part of the talk, where I would like to point out that the ciliates are obviously very successful in anoxia, or inhabiting these harsh environments, So we need to we want to know more about why are they so successful, and how are they dealing with the anoxia, and how are they producing energy without oxygen, you know. So if we look at the classical aerobic mitochondria, you see there these cristae, these invaginations within the membranes that are hosting, or that are space where the oxidative phosphorylation and major parts of energetic metabolism are happening. And if you look at the other anaerobic mitochondria, or mitochondria related organelles, as we call them, in anaerobic protists, or anaerobic eukaryotes in general, you can see that the cristae are slowly getting lost. You see, if you look at the picture on the right here, the sort of textbook mitochondrion, over time, or during the evolution and during the transition to anaerobiosis, the cristae are getting lost, and so is the space for the complexes, or for the metabolic enzymes that are helping to produce energy via utilizing oxygen. And here on the right, you can see the mitochondrion-related organelle of one of the anaerobic ciliate, the Muranotrichea, and you see that there are almost no visible cristae. I'm sorry. And so, yet, if we look at their metabolism, we will find out something that one wouldn't expect. Here you see the usual typical aerobic energetic metabolism that's happening in mitochondria, and you see the different complexes, and I will definitely not bore you with like the details of what is happening there, but I just want you to know that you know, the oxygen is key molecule there, and during the electron transport chain, there are various oxidative and reductive reactions going on, and finally there is the ATP synthase pump that's thanks to the oxidative phosphorylation usually is producing ATP, so a form of energy. And if we look at the situation in the anaerobic metabolism, usually in very strict anaerobes or generally anaerobic protists, we will see that most of those complexes are completely missing, sometimes to the point where there are none. And recently, in 2016, a team of Anna Karnkowska has discovered that there are actually protists that have no mitochondria whatsoever. They're called monocercomonoides, and I think they're inhabiting chinchillas. But in the case of the ciliates, the mitochondrion actually has remained relatively complex, and the ciliates have retained several of the complexes that are playing a role in the production of energy and different processes that are usually happening in the cell. And you can see that that only one of them, the Muranotrichea, has retained complete complex 5, or otherwise known also as this ATP synthase. So they're likely able to produce energy the same way as most aerobes do, which is interesting because you know, ciliates are in the protist world, for protistologists, they're relatively large and complex organisms. because usually anaerobic organisms are very reduced and kind of tiny. So this is a unique case of complex organisms being able to maintain and produce enough energy while keeping their active lifestyle. So we can think of what's their secret to being able to diversify within anoxia and keep their cells large and active. And one of the things we could think about are also some pre-adaptations in form of maybe different iron hydrogenases that have been discovered in most of the anaerobic ciliates, and that has led some authors to think that maybe all of the ciliates and maybe the ancestor of ciliates had some sort of metabolic or genetic adaptation to be able to withstand longer periods of anoxia. Sorry. And if we look at the phylogenetic or phylogenomic tree that was based on over 120 genes, you will see that there is actually one major clade of anaerobes here in the cell supergroup. We have called it APM clade, and it's including the Metapida or Armaphoria and some endobiotic organisms like Clevelandellida, and also the Muranotrichea and Parablepharisma. And if we include if we perform single gene analysis to include organisms that we have no genomic data for, such as the deep sea Caryocotrichea, we will see that they might also be related to Muranotrichea and Parablepharisma, and there is also this other group, Odontostomata. So basically, many of the known anaerobic lineages within ciliates are forming this one clade that had a common ancestor, and that's quite that gives us a great opportunity to study the evolution, because there are different levels of reduction, different levels of the tolerance to oxygen, and being able to cultivate routinely the Parablepharisma and Muranotrichea that might be related to these enigmatic deep sea organisms that no one has ever seen alive, and they have only been isolated from the deep sea as fixed organisms, and it was difficult, I think, to even obtain enough DNA for the single gene analysis. So that gives us more means to study the evolution of even these enigmatic organisms, and also about the evolutionary ways of the transition to the anaerobiosis, and in particularly the transition to the obligate anaerobiosis, because we know a lot about microaerophils, or about these like facultative anaerobes, but we don't know much about the reasons that are keeping the organisms in the anoxic environment. And so we think that in the ciliates, this may have been facilitated actually by the symbionts. And I have this horrible animation that I've tried yesterday, so please excuse the quality. But it's supposed to show that even aerobic ciliates are known to travel to anoxia or to hypoxic sediments to feed on these anaerobic bacteria, and when they do so, sometimes I guess some of the methanogens or some of the bacteria that they feed on have escaped the digestion and they have actually become endosymbionts. And we think that by providing the host with boosted metabolism rate, which has been proved already many years ago, that they're actually helping the ciliate to increase the rate of their metabolism, that they might have motivated the ciliate or helped the ciliates to gain this advantage among other anaerobic protists or anaerobes in general by helping the metabolism, because in many anaerobes or in anoxia, usually the metabolism is slower and has lower yields of energy for obvious reasons. And so then the symbionts might have likely caused the ciliate to stay, because if he would come back to the oxygenated waters, he would risk killing the symbiont or losing the symbiont, and potentially endangering himself too. So over time, over the evolution, they have lost like metabolic capability to metabolize oxygen or to use the aerobic metabolism, and they had to permanently reside in the anoxia. And they couldn't come back. And so to kind of wrap it up, I want us to come back to see that to see how many factors are impacting today's ocean and the changes that are going on in the ocean. And I would like for us to as sort of take-home message to realize that although the free-living prokaryotes, archaea, and bacteria are having obviously major roles on the biogeochemical cycles and on the carbon cycling and also on the climate change and greenhouse gases emissions, we should keep in mind that many of these prokaryotes are actually in some sort of symbiosis with other organisms, maybe also some of them anaerobic ciliates. And so to sort of conclude it, we know that ciliates are very successful in anoxia, and they were able to maintain the large cells, and they're able to produce enough energy and keep their metabolism very effective. And their symbionts might play a large role in this. And so they could serve as a great model group to study generally life in anoxia and adaptations to these conditions. And if you wanted to learn Czech and read more about anaerobic protists, we were lucky enough to get the attention from media, so we had quite some funny articles or TV shows about ciliates. So thank you for listening, and I would like to also thank huge time to Roxanne for hiring me as a postdoc during these crazy times, and also of course to all of the people that we have been collaborating on this, and of course my supervisor from Charles University that has introduced me to the world of protists and anaerobic ciliates, and of course to all of the funding, and also to Bio at Noon and GSO for letting me be here. Thank you.
Thank you very much, Hannah. That was beautiful. There were some amazing pictures in there. An amazing amount of work you've done too. Can you see the chat? There are a few questions there, but I can read them to you as well.
I should be able. Give me a. I'm now suddenly like completely useless with my computer, and I cannot find it.
Okay, I will read.
Oh, here.
You got it? Okay.
Yeah. Sorry.
No worries.
Yeah. That's a good question about the 30 meters deep sediments. So it's just a sediment on the bottom of a shallow sea. So 30 meters deep in the water. Well, let me rephrase it. The sea there was only 30 meters deep, and we have sampled the sediment at the bottom. Yeah, I hope that answers.
Thank you.
Does the symbiont, does the same ciliate species always have the same symbiont, or does that change too? That's also a great question, and we have actually been trying to find an answer to it, but we don't have a clear answer yet, and we're trying to find out through doing these experiments where we actually mix cultures of different ciliate species together and let them coexist for several months. And then we were sequencing the symbionts before the experiment and then after the months of the experiment to see if they might have switched the symbiont, at least in one of them or so. And so far we haven't seen that. And maybe it's just that, several months is not long enough period, or maybe that this particular species is not able to do this. So definitely we need to do a lot more of these experiments on a broader scale, and also try to find some other options how to find it. But so from looking at the genomes, it looks like the symbiosis might be relatively recent in some cases of the freshwater Heterometopus paliformis, but in some other cases that in a study that was published by another team, Andreas Lind et al. From ETEMA, from Swedish Uppsala University, in a marine Metopus contortus, the genome was a little bit more reduced, and it's possible that basically different symbioses in different species were established on different evolutionary scale or during different time period. Obviously, that we think that the symbionts have switched over the evolution, that some have lost symbionts and gained another one, and so on. Yeah.
Great, thank you.
And are these ciliates found as part of pelagic marine snow aggregates, perhaps in anoxic microhabitats within particles? I'm not sure, and it's another student of rock science, Anna Schreckingust, who I believe is here as well. She's currently looking at like environmental data that could tell us where everywhere we can find anaerobic ciliates, and so we are looking at she's looking at deep sea data or shallow waters data sets of sequences from environmental data from these. So I'm not quite sure about any particular study on this. Maybe Roxanne or Anna would know more if you want to.
It's really interesting because I hadn't thought of including or looking for like marine snow sequencing data sets. And that's a really interesting question. I don't know a lot about that, and I don't know if they're out there, but I think it's definitely a good thing to look for.
Yeah. Thank you for Karen for pointing that out. Yeah. Anyone else has any question or comments or criticism?
Hi, this is Tatiana Reinierson. And I wanted to congratulate you for fitting in the first and possibly the last use of the word chinchilla at noon. I haven't heard that before. And you mentioned that the symbionts are spread across the phylogenetic tree of the methanogens. So I'm wondering, and you mentioned this experiment where you tried to sort of cross-fertilize the symbionts. Do you have any sense of, from your experiments and reading, if the symbionts are, if this is a very close sort of strain-strain kind of interaction, or if there's a competition that, one bacterium.
Okay, are we done? Okay.
If one bacterium is a better competitor to be colonizing the surface of the ciliate, like, do you have an idea of maybe what some of the underlying dynamics might be yet?
Yeah, that's a great question. We're we don't have like a clear pattern yet, but also from the literature and from some of our recent data, we see that there are obviously certain species or genera or lineages of the methanogens that are more keen to establish the symbiosis. Not like one group or one clade. There are many of them, but for example, the methanobacterium seems to be able to have established relationships with many different unrelated ciliates, and but those are mostly on the inside of the cell, and then on the surface of the cell they're mostly the sulfate reducing bacteria, which actually I have no information about their host specificity yet, and I would also like to find out about it. So hopefully I'll be able to give you a better answer during the next year or couple of years, or hopefully less. Yeah, sorry. Or did I maybe I have missed something from the question?
Nope. Thank you.
Yeah, thank you.
Johana, I have a question about the ectosymbionts. Does that require that the host be in an anaerobic environment, or do the symbionts have a way of making some sort of micro zone around them to keep away from the oxygen?
Yeah, that's I keep getting these awesome questions. Thank you. Yeah, this is something that we were trying to also see during the analysis of the transcriptomes or the metabolic parts. And so ciliates, as many other eukaryotes, they have protective means or protective mechanisms from oxygen and alternative oxidases, but I think that it's more like vice versa, that the symbionts have obtained the strictly anaerobic symbionts while traveling to anoxic sediments. And so although we know that from some culture experiments and so on, we know that and also from the environment, I mean, from the studies, we know that the ciliates, even the anaerobic ciliates, can travel to aerobic environment for short periods of time, or they can survive exposure to low levels of oxygen. And obviously their symbionts survived it, and the ciliates survived it, but obviously they could not permanently, go back to aerobic environment and retain the symbionts there. I don't know if any ciliates have tried that in the past, but they have not made it to the present, so they were probably not so successful. So, does that answer that, or?
Yeah, thanks.
Yeah, thank you. Thank you. I'm seeing another new in the discussion. If the capacity David mentioned, there would be a means to colonize marine snow particles. Yeah, that's a good point.
Well, thank you very much, Johana. If people want to unmute and give a round of applause, that would be great. And postdocs and grad students, please stay on and we'll continue the discussion. So thank you, Johana.
Thank you all as well for doing this.